Lab 2, topics enzymes and reaction rates. So, start with a little review on enzymes. What is an enzyme? A catalyst, B, a protein, C, a lipid, D, A and B only, or E, A, B, and C? Well, it turns out it is A and B. Enzymes are catalysts and enzymes are proteins. Proteins and catalysts. So, enzymes are proteins that serve the function of catalysts. There are many catalysts out there in the world, but the ones we're most concerned about are the enzymes that function in the human body. So, many catalysts exist that are not proteins. We are going to focus on enzymes, which are catalysts that are proteins. So, in general, catalysts are chemicals that increase the rate of reaction and are not changed by the reaction, so they can get used again and again and again. So that is pretty amazing. And overall, they don't change the nature of the reaction. Same substrates, same products, everything is occurring as expected. It's just happening faster. So basically, we have this lovely graph here. Here is, say, your substrate, and then here is the product. In this case, we're breaking up sucrose into its components, glucose and fructose. And then normally, without an enzyme, there's this huge mountain of activation energy needed. And because of this, sucrose only breaks down into fructose and glucose, say, once every 1,000 years. Well, that is way too slow for the human body. So we have lovely enzymes that lowers the activation energy to a manageable level. So now we can break sucrose into its components in milliseconds so that we are able to speed up the chemical reaction by decreasing the activation energy, getting things done at the speed of life. All right, enzymes. Enzymes have a special area called an active site. The active site is a very specific shape, the shape that will fit one kind of substrate. So here's a substrate, here's the active site, fits in just correctly into the enzyme, giving us the enzyme substrate complex. This kind of specificity is important. We want the enzyme to only function on its substrates. And this is often called a lock and key mechanism because the substrate acts like a key. Key has to be a certain shape. Enzyme acts as a lock. It is also a certain shape. So to get things to happen, you have to have the correct shaped key fitting into the correct lock. Here's another example. We have these two substrates. They fit into the enzyme's active sites, giving us the enzyme substrate complex. And then the reaction occurs and it releases the products. All right, most enzymes, the name of the enzyme ends with the suffix ase. So if you see a word ends in ase, that is an enzyme. So for instance, phosphatase, ase, it's an enzyme that removes phosphate groups. Synthetase, ase, it is an enzyme that's involved in dehydration synthesis. Amylases, ase, they are enzymes that break down carbohydrates. So remember, ase, you see it at the end of the word, and that is talking about an enzyme. All right, here's a lovely chemical reaction. What in this chemical reaction is the enzyme? What in this chemical reaction is the product or products? What in this chemical reaction is the substrate or substrates? Need more time? Feel free to pause the video. And the answers are, boom, sucrase is the enzyme. You see it's on both sides because it is not used up or changed by the chemical reaction. It's the same before and after the chemical reaction. The products are glucose and fructose. The arrow points to the products. Again, the arrow points to the products. If you haven't learned this fact by now, well, that's sad. The arrow points to the products. And that means what's on the beginning start side is the substrate. Sucrose and water are the substrates. Another way this chemical reaction could be written would be sucrose plus water yields glucose and fructose, and sucrase is just over the arrow rather than showing up on both sides. All right, there are things that affect the speed of the enzyme. So the enzyme's activity is affected by a lot of different things, such as temperature, such as pH, such as concentration of substrates, such as concentration of the enzyme. So we're going to talk about that. So here is temperature. We'll start with the temperature. We have this lovely graph here. As you can see, on one side, it's showing enzyme activity. So the further toward the top, the faster the enzyme is working, the reaction rate is faster. And then this line, the x-axis, is showing the temperature. Temperature increasing as we go to the end of the chart from left to right. So here is the graph of the activity. Here's a random question. At what temperature would you expect human enzymes to function the best at? Human enzymes. Well, it would be body temperature, of course. And as we can see here, body temperature is about 37 degrees Celsius. So human enzymes function best at 37 degrees Celsius. Now, if you're lower than that, the enzyme is less active. If it's higher than that, the enzyme is less active. So what you see is that as you increase the temperature, you will increase the activity of the enzyme until you reach the ideal temperature, which in humans is about 37 degrees Celsius. And then if you continue to increase the temperature, the enzyme activity will decrease. And if you take temperature up high enough, you will denature the enzyme and it'll become inactive and no longer functional. So again, temperature functions best at the ideal temperature. It will increase in activity as it increases with temperature until it reaches the ideal temperature. Then continuing increasing the temperature will decrease the activity of the enzyme until if you increase the temperature enough, you will denature the enzyme, making it permanently inactive. And it's permanent. You cannot undenature proteins. You cannot unfry the egg. All right, pH. pH is a little similar. Enzymes function in a narrow range of pHs. pH is way too low, the protein becomes non-functional and denatured. The pH is way too high, the protein becomes non-functional and denatured. So there's a certain range that enzymes can function at. The ideal pH, of course, reflects the environment that the enzyme is in when it normally functions in the human body, which means that the stomach's environment is different from the mouth, is different from the small intestine, so the ideal pH for enzymes in these three locations will be different. So for instance, in our lovely chart here, we see that we have the enzyme pepsin. Pepsin's found in the stomach. Pepsin likes a nice acidic environment of around pH 2. So as you increase the pH, the activity of the enzyme will increase until you reach the ideal pH. If you continue increasing the pH, the activity of the enzyme will decrease, and if you go to extremes, the enzyme will stop functioning. Salivary amylase is found in the mouth. Its ideal pH is around 7. And again, as you increase the pH, the enzyme becomes more active until you reach the ideal pH. Continue to increase the pH and the enzyme will become less active. Go to extremes and you denature the enzyme. And finally, trypsin here. Trypsin works in the small intestine. Small intestine is basic in nature, so trypsin works best at a pH around 9. So again, as you increase the pH, you increase the activity of the trypsin enzyme until you reach the ideal pH of around 9. Continue to increase the pH and the trypsin enzyme becomes less active. Go to extreme pHs and you denature the enzyme. All right, moving on. Amount of substrate. So obviously, if there's no substrate, there is no reaction. And it turns out the rate of the reaction will increase, so it gets faster as you increase the amount of substrate. However, this plateaus out so that you eventually get to a point where the substrate is saturated, and adding more substrate will not increase the reaction rate. Basic idea is, say you have 3 enzymes. Enzymes are like robots, do only one thing. Well, you have a little bit of substrate, then you've got only one of the robots active, so the reaction rate is a certain level. Add even more substrate, now you got all the robots, all the enzymes active, so it is active, the reaction rate is as fast as it can get. Add even more substrate, and you will not get an increase in activity because you're using up all the enzymes already. They're already working as hard and fast as they can. More substrate does not get more of them working. All right, another question is the effect of the amount of enzyme. Well, it turns out that as you increase the amount of the enzyme, you increase the reaction rate as long as you have plenty of substrate. So more enzyme means a faster reaction rate as long as you have plenty of substrate. If you run out of substrate, you run out of activity. So one enzyme acting on the substrate goes at a certain speed. 3 enzymes acting on the substrate, 3 times as fast. 5 enzymes acting on the substrate, now it's even faster, and so on as long as you have plenty of substrate. Cofactors and coenzymes. Cofactors and coenzymes, are structures, molecules needed by some enzymes to function. So some enzymes need cofactors, some enzymes need coenzymes. Not all enzymes, but some of them. Cofactors are inorganic, so they're usually a mineral. For instance, the iron in hemoglobin is a cofactor for the hemoglobin. Coenzymes are often vitamins and organic in nature. So basically, you need enough cofactor or coenzymes for the total amount of enzymes. Once you have more than that, you've saturated the system. So too little, slow reaction rate; too much, no overall increase. So here you have enzymes with no cofactors. They can't do anything. Now you have enough cofactor for, say, one enzyme, it goes at a certain rate. Enough cofactor for 2, now it's even faster. Enough for 3, it's even more faster. You add even more cofactor though, and it's saturated now. All the enzymes have cofactors. Adding more cofactors will not help things out. All right, so in summary, enzymes activity can be measured, and this is the rate of the reaction, or basically the speed. It is how much substrate is produced over a specific amount of time. And this can be influenced by temperature, with the fastest rate is at the ideal temperature. Too high or too low will not be as fast. pH, fastest rate for pH is at the ideal pH. Too high or too low will not be as fast. Concentrations of substrates, adding more substrate will speed things up until the substrate is saturated. And concentration of enzyme. Add more enzyme, this will speed up the reaction rate as long as you have plenty of substrate. So again, these are various factors that affect the reaction rate, aka affect the amount of substrate produced over a specific amount of time.